population genetics of ecological replicates, feather lice, pigeons.
Okay. So natural populations are typically subdivided or structured because geographic or ecological factors prevent individuals in the population from randomly mating. So when this happens, groups of individuals in the population can diverge from one another and genetically differentiate. And this is, of course, important, the degree of structure, because this affects rates of local adaptation, speciation, as well as extinction. So with my thesis, I'm interested in how do ecological factors influence population genetic structure. So I want to look at things like abiotic factors, such as climate patterns, as well as biotic factors, such as dispersal and competition, and how this influences genetic diversity within a species. And for this talk, I'm going to focus on one particular mode of dispersal. So I'm using feather lice. So here's a picture of a feather louse. It's a small wingless insect, and here you can see it's crawling on the feathers of its bird host. These small insects are adapted to life on their host. So feather lice are permanent host-specific parasites. So they started as an egg, go through 3 level instars, and then go to an adult form, and this happens all on their bird host. So here you can see pictures of 2 feathers. So the lice eat the feathers. So on the left you see an intact feather, and on the right you see a feather that has been mostly eaten up by these feather lice. So to answer my question, I'm looking at 2 species of louse and their pigeon. And these 2 species are very host-specific. They're only specific to this pigeon, the Columba livia. So I'm looking at wing lice, which are kind of long and slender, and they are adapted for crawling and inserting into the wing feathers of the host, whereas body lice are more adapted for crawling on the body and the feathers of the host on the body. So we consider this system to be an ecological replicate. We think these 2 species are ecological replicates because they live in the same exact environment, the same host. They have really similar life cycles. They have almost the same generation time, and they have really similar modes of dispersal. Typically, the most common, modes of transmission. So typically, these parasites transmit from mother to offspring, but they also can engage in horizontal transmission. So wing lice, however, only wing lice and not body lice, can attach to flies and phoretically transmit to other pigeons. So these flies are also parasites of the pigeons. So they fly to a pigeon, hop on it, and take the blood meal. And during that time, the wing lice can attach to that fly and potentially go with the fly where it goes. So if it goes to another pigeon host, the wing lice can go with it and potentially transfer it to another host. So as I said before, only wing lice can do this and not body lice. So these wing lice have an additional mode of horizontal transmission, which the body lice do not. So my question is, what is the influence of phoretic dispersal on population genetic structure? And you may say, oh, that seems pretty specific to boring pigeons and lice, but it's not. Phoresis is actually really common. We can see here a picture of mites, phoretic on a beetle, and so forth. So back to this question, what is the influence of phoretic dispersal on genetic structure? So to answer this question, of course, first I have to go out and sample. So we sample these really glamorous rock pigeons, which can even be found here on the beach. And to do this, I go to the urban jungle and set up ladders, and usually the cops come because I look pretty sketchy, but that's okay. So I just use a variety to catch these birds. I fumigate the birds for all their parasites. So I use Ecological to kill all the parasites and collectible parasites. And so far I have a wide geographic sampling across the US, but today I'm going to talk about data from 2 particular points, Salt Lake City, Utah, and New Orleans, Louisiana. So I developed 10 new microsatellite loci for both louse species, and these are highly polymorphic. And I wanted to use microsatellite loci because they're good markers for understanding the recent past or ecological time, which I'm interested in. So here I'm only showing a little bit of data. I hope to have more for this meeting, but this is what I have now. So what I did was I took 3 birds from each location, and I genotyped 10 wing lice and 10 body lice from each bird. And so right now I have the data for just 4 of the microsatellite loci. And just a quick review before I run into the results. Talking about genetic differentiation, I measured it in terms of FST. So we have 2 circles here. On the left would be a subpopulation, and on the right another subpopulation. And blue and green lice, which are represented there, would represent different alleles. So in these 2 populations, 2 subpopulations that you see, the alleles are pretty spread between the 2, so FST would be 0. And then of course, when one allele is fixed in a subpopulation and the other, then the FST would be 1. So first I'm going to show you my sampling that I did from Salt Lake. So here you can see a diagram, it's a highlight diagram, and I sampled 3 different flocks. So these flocks are about 6 miles and 4 miles from each other. And what I did was I looked at 1 bird within each flock, 10 wing lice and 10 body lice on each bird in each flock. And we can see that for body lice, between flocks, there is a moderate amount of genetic differentiation. But for wing lice, there's not. So I think because that the wing lice can hop on the flies and have this extra mode of dispersal, I think that's why we're seeing that genetic differentiation, because body lice have a reduced dispersal ability. So body lice show more population structure than wing lice between different bird flocks. Whereas in New Orleans, I took birds from a single flock, so that all live together in the same area, and I compared them. And we do see that for body lice, we see significant structure, although it's less than between flocks, whereas for wing lice, we do not. And so body lice show more population structure than wing lice, even on individual birds. So we have this hierarchical sampling, and we're seeing this between we're seeing this differentiation in lice between bird flocks as well as between individual birds. So to summarize that, I think phoretic dispersal decreases population structure. And so here you have a map of North America, and on the dotted line, below the dotted line, there are areas where that vector, the hippobossid flies, occurs. So this whole area is where this particular avian vector occurs. Whereas above this line, these hippobossid flies do not occur. So the wing lice would not have a chance to disperse as much on the hippobossid flies. So what I'm doing now is comparing my US geographic sampling and getting this Canadian sampling to compare areas with flies and without flies and looking at the lab structure there. So what I found was that in areas with flies, we have low structure with these wing lice species, high structure with these body lice species, and I'm working on this next part. So thanks for sticking around for the last talk.
Have you sampled hippobossid flies as well from pigeons?
Yes.
So you're going to also analyse those?
I can. So I'm working on getting the host genotypic data as well, so hopefully I'll have time to do the flies as well. But.
What do you know about how far they disperse? Because my experience on this is that they don't look terribly well adapted to the long distance dispersal of those flies.
The flies?
Yeah, they tend to leap off burn stuff into your plates when they're not.
Yeah, I don't know for sure.
Flies. Yeah.
But I know that, so the flies can stay on the pigeons, and the pigeons can go up to 200 miles. So potentially during that time, the fly could be on the pigeon. Because I know that they really bury themselves in the feathers of the pigeon. So I think that would be the mode between different flocks in different areas. Yes?
Relative to that question, where are the larvae of hippobossid?
I'm sorry, what?
Where do the larvae of hippobossid flies develop? On the pigeon or elsewhere?
No, the larvae do not develop on the pigeon. We think they develop in the nests. So, yeah, we think they develop in the pigeon nests.
I'm curious in terms of, is the only mode of transmission by the flies or also by bodily contact among the pigeons?
So in close contact, we can get transmission, like between mates and such. Mhm.
And how would that actually affect body versus wing?
Yeah, that's a good question. Don't know for sure. But as I see structure, even on host individuals, it looks like maybe wing lice potentially can transfer more, but I'm not sure.
I was just curious about the kind of densities of the parasites on the flies for dispersal, where and if there's any sort of, if you've seen any geographic variation in that as you go towards that cut-off point.
Well, I mean, just as an anecdote, I was on the beach inspecting a pigeon here, and there was like over 40 flies that were on that particular bird.
Wow.
Whereas in Salt Lake, the prevalence of flies, we're not exactly sure, but typically we only see 1 to 2 flies on the bird. So there is some variation, I think.